Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel Diversity of Plants Zone. In this channel you will get every necessary information related to the plants and their diversity throughout the world. Before starting this video, I request you all to please subscribe, share and like my video. My today's video is about the most important topic, Domain Archaea. I hope you will enjoy this video. So let's start the video. First of all, I will discuss about introduction to Archaea. Archaea are a domain of single-celled microorganisms. They have no cell nucleus or any other organelles inside their cells. In the past, Archaea were classified as an unusual group of bacteria and named Archaebacteria. But since the Archaea have an independent evolutionary history and manifest numerous differences in their biochemistry from other forms of life, they are now considered as a separate domain in the three-domain system. In this system, the three primary branches of evolutionary descent are the Archaea, Eukarya, and Bacteria. Archaea are further divided into four recognized phyla, although other phyla may exist. Of these groups, the Crenarchaeota and the Eukaryotic are the most intensively studied. Classifying the Archaea is somewhat challenging, since the vast majority have never been studied and have chiefly been detected by analysis of their nucleic acids in samples from the environment. Archaea replicate asexually in a process which is known as binary fission. Archaea achieve a swimming motility via one or more tail-like flagellae. Many archaeans are extremophiles, achieving wide environmental tolerance of temperature, salinity, and even radioactive environments. So Archaea are thought to be significant in global geochemical cycling, since they comprise an estimated 20% of the world's biomass. However, very little is known about the domain, especially marine and deep-sea benthic varieties. Now the question is, what are Archaea? Archaea are a group of microorganisms that were discovered in the early 1970s. Like bacteria, they are single-celled prokaryotes. Archaeans were originally thought to be bacteria until DNA analysis showed that they are different organisms. In fact, they are so different that the discovery prompted scientists to come up with a new system for classifying life. There is still much about Archaeans that is not known. What we do know is that many are extreme organisms that live and thrive under some of the most extreme conditions, such as extremely hot, acidic, or alkaline environments. Next, I am going to discuss about the definition of Archaea. Archaea are unicellular organisms that make up the third domain of organisms on Earth. As such, they are different from the other two domains that include bacteria and eukaryota. Like bacteria, however, Archaea are prokaryotes that share certain characteristics with bacteria. This is one of the reasons Archaea were previously thought to be a type of bacteria. Due to their ability to survive extreme conditions, and they can be found in a wide variety of environments, ranging from the lakes and soil to the Dead Sea and the deepest parts of the ocean floor. So some examples include Aeropyrum, Pernix, Thermocephera aggregans, Ignisphere aggregans, Cephalobus tocodi, Metallosphere sedula. And the next is Staphylothyrmus marinus and Thermoproteus tenax. So this is archaea which is growing in the runoff of the Giza as shown into the figure. Next I am going to discuss about the archaea cells. So archaeans are extremely small microbes that must be viewed under an electron microscope to identify their characteristics. Like bacteria, they come in a variety of shapes including coxy, round, bacilli, rod-shaped, and irregular shapes. Archaeans have a typical prokaryotic cell anatomy: plasmid DNA, cell wall, cell membrane, cytoplasm, and ribosomes. Some archaeans also have long, whip-like protrusions, which is called flagella, which aid in movement. So next I am going to discuss further about the domain Archaea. Archaea, domain Archaea, any of a group of single-celled prokaryotic organisms, that is organisms whose cells lack a defined nucleus, that have distinct molecular characteristics separating them from bacteria, the other more prominent group of prokaryotes, as well as from eukaryotes, organisms including plants and animals whose cells contain a defined nucleus. So this is archaea as shown into the figure. Archaea is derived from the Greek word archaeos, meaning ancient or primitive, and indeed some archaea exhibit characteristics worthy of that name. So members of archaea include Pyrolobus fumeri, which holds the upper temperature limit for life at 113 degrees Celsius and 235 Fahrenheit, and was found living in hydrothermal vents. Species of the Picrophilus, which were isolated from acidic soils in Japan and are the most acid-tolerant organisms known, capable of growth at round pH 0, and the methanogens which produce methane gas as a metabolic byproduct and are found in anaerobic environments, such as in marshes, hot springs, and the guts of animals, including humans. In some systems, for classifying all of life, the archaea constitute one of the three great domains of the living creatures. In 1977, American microbiologist Carl Voss, on the basis of analysis of ribosomal RNA, proposed that the prokaryotes, long considered to be a single group of organisms, essentially the bacteria, actually consist of two separate lineages. So Voss called these two lineages the eubacteria and the archaebacteria. These names were subsequently changed to bacteria and archaea, the archaea being distinctly different from bacteria. But Voss' splitting of the prokaryotes into two groups has remained, and all living organisms are now considered by many biologists to fall into one of three great domains, which are archaea, bacteria, and eukarya. Further molecular analysis has showed that the domain Archaea consists of two major subdivisions, the Crenarchaeota and the Euryarchaeota, and the minor ancient lineage, which is called as Corarchaeota. So other subdivisions have been proposed, including the Nanoarchaeota and Thymarchaeota. So now I will discuss further about the domain Archaea. Organisms are now classified into three domains and 6 kingdoms. The domains include Eukaryota, Eubacteria, and Archaea. Under the Archaea domain, there are three main divisions or phyla. They are Crenarchaeota, Eukaryotic, and prokaryotic. So first I will discuss about the Crenarchaeota. Crenarchaeota consists mostly of hyperthermophiles and thermoacidophiles. Hyperthermophilic organisms or microorganisms live in extremely hot or cold environments. On the other hand, thermoacidophiles are microscopic organisms that live in extremely hot and acidic environments. Their habitats have a pH between 5 and 1. So you would find these organisms in hydrothermal vents and hot springs. Crenarchaeota species. So examples of Crenarchaeotans include. So first example is Cephalobus acidocladrius. Cephalobus acidocladrius, which is found near volcanic environments in hot and acidic springs containing the sulfur. And the next example is the Pyrolobus fumari, that live in temperatures between 90 and 113 degrees Celsius. Next I am going to discuss about Eukaryotic. So Eukaryotic organisms consist mostly of extreme halophiles and methanogens. Extreme halophilic organisms live in salty habitats. They need salty environments to survive, and you would find these organisms in salt lakes or areas where the seawater has evaporated. Methanogens require oxygen free environment, which is called anaerobic conditions, in order to survive. They produce methane gas as a byproduct of metabolism. So you would find these organisms in environments such as swamps, wetlands, ice lakes, and the guts of animals, for example cow, deer, humans, and in sewage. Next is the Eukaryotic species. So the examples of Eukaryotes include, first is Halobacterium. Include the several species of halophilic organisms that are found in salt lakes and the high saline ocean environments. Next is the Methanococcus. So Methanococcus genesiae was the first genetically sequenced archaean. This methanogen survives or lives near the hydrothermal vents. Next is the Methanococcoids, Botoni. So these psychrophilic, cold-loving and methanogens were discovered in the Antarctic and can survive extremely cold temperatures. Next I am going to discuss about the Prokaryotic. Prokaryotic organisms are thought to be very primitive life forms. Little is currently known about the major characteristics of these organisms, and we do know that they are thermophilic and have been found in hot springs and obsidian pools. So next I am going to discuss further about the Domain Archaea and Archaebacteria. In the 1970s, while studying the relationships among prokaryotes using DNA sequences, a scientist named Carl Voss discovered some unusual organisms that appear to be very distinct from prokaryotes and eukaryotes. These seemingly unusual organisms were neither prokaryotes nor eukaryotes. The extreme difference in the genetic and molecular levels lead the scientists to the discovery of the third domain of life, which is called the Domain Archaea. So the members of the Domain Archaea, or simply Archaeans, are incredibly abundant in environments that are hostile to all other life forms. Hence their place among the other living organisms long went unrecognized. The Domain Archaea is such a remarkably diverse and successful clade of organisms Next I am going to discuss about the domain and the kingdom of Archaea. The domain and kingdom of Archaea. So Archaea share characteristics of both prokaryotes and eukaryotes, which is why they exist on a separate branch between bacteria and eukaryotes in the phylogenetic tree of life. So when scientists discovered that archaebacteria were not actually ancient bacteria, they renamed them Archaea. The following features define the Archaea single-cell organisms. They are prokaryotic cells but are genetically more like eukaryotes. Cellular membranes consist of branched hydrocarbon chains, unlike bacteria and eukarya, connected to the glycerol by ethylene linkage. So Archaea cell walls have no peptidoglycans, polymers made up of sugars and amino acids that form a webbed layer outside the cell walls of the most bacteria. So when Archaea do not respond to some antibiotics that bacteria react to, they do react to some antibiotics that distress the eukaryotes. Archaea contain ribosomal ribonucleic acid, ribosomal RNA, specific to the archaea, essential for protein synthesis, identified by the molecular areas, noticeably unlike that of the ribosomal RNA found in bacteria and eukarya. The main classifications of Archaea include the Crenarchaeota, Euryarchaeota, and Corarchaeota, well as proposed subdivisions of Nanoarchaeota and the proposed Thymarchaeota. Individual classifications indicate the types of environments in which researchers and the scientists find these single-celled organisms. Crenarchaeota live in environments of extreme acidity and temperature and oxidize ammonia. Euryarchaeota include organisms that oxidize methane and love salt in deep sea environments. Other Euryarchaeota that produce methane as a waste product, and Corarchaeota, a category of archaea that also live in high temperature environments. Nanoarchaeota differ from other archaea in that they live atop an other archaean organism, which is called as ignicoccus. So subtypes of Corarchaeota and Nanoarchaeota include methanogens, organisms that produce methane gas as a byproduct of the digestive or energy-making processes, halophiles or salt-loving archaea, thermophiles, organisms that thrive in extremely high temperatures, and psychrophiles, archaea, organisms that live in extremely cold temperatures. So next I am going to discuss about the history of Archaea. Before its separation, the Domain Archaea was formerly called the Domain Archaebacteria, which is a misnomer, since these organisms were not bacteria but belonged to a separate clade. The name Archaebacteria was previously an untested hypothesis about the evolutionary status of the organisms, having a type of metabolic ability that seems to be highly suited to the primordial conditions of life on Earth. Therefore, there is a substantial possibility that this new group of organisms is the oldest. The three domains of life are illustrated below, showing each of the domains is very distinct from the others. So, there are three domains. So, first domain is the bacteria, which includes the cyanobacteria or heterotrophic bacteria. So, next domain is the eukaryota, which includes fungi, animals, plants, grow mist, alveolates and rhodophytes, and flagellates and basal protists are included in it. Third domain is the archaea, that includes halophiles and thermophiles as shown into the figure. So next I am going to discuss about the archaea phylogeny. Archaea are interesting organisms in that they have genes that are similar to both bacteria and eukaryotes. Phylogenetically speaking, the Archean bacteria are thought to have developed separately from a common ancestor. So eukaryotes are believed to have branched off from Archeans millions of years later. This suggests that the Archeans are more closely related to the eukaryotes than bacteria. So this is the tree of life, which is according to the three domain system. So first, Archaea is divided into the three phyla. First is Eukaryotica, Prenarchaeota and Corarchaeota. On the other hand, Eukaryota is divided into the plants. So plants are further divided into non-vascular and vascular. So vascular are further divided into the seedless plants and with seed plants. So seedless plants are with seed plants are further divided into the gymnosperms and angiosperms. On the other hand, animals are divided into the cnidarians and bilaterals. Bilaterals are further divided into the arthropods, and arthropods are further divided into the crustaceans and arachnids and myriapods and insects. On the other hand, the bilateral is further divided into the vertebrates, and these vertebrates are further divided into the bony fishes. Next is amphibians, because tetrapods are further divided into amphibians and amniotes. And amniotes are further divided into mammals, and the mammals are divided into placentals, marsupials and monotremes. So amniotes are further divided into the birds and reptiles, which are further divided into the turtles, lizards and snakes. And vertebrates are further divided into cartilaginous fish. Bilateral are also divided to form mollusks. Next is the fungi. So fungi is further divided into basidiomycetes, zygomycetes and ascomycetes or ascomycetes. And the eukaryota are further divided into protista. So the third domain, bacteria, is divided into the Vibrio, and next is the spirillum. This is bacillus and this is cockles as shown into the figure. So this is the tree of life, which is according to the three domain system as shown into the figure. Next I am going to discuss about the evolution. So very early probable prokaryotic cell fossils have been dated at approximately 3.5 billion years before the present day, making them some of the most primitive and ancient life forms on the earth. However, prokaryotes generally lack the distinctive morphologies. Thus, the fossil shapes cannot be used to identify them as archaea. Instead, chemical fossils of unique lipids hold greater information, since such compounds do not occur in other organisms. Some research indicates archean or eukaryotic lipid remains are present in the shells as old as 2.7 billion years. Such lipids have been identified in the Precambrian formations, the earliest of which are present in the Isua greenstone belt of the western Greenland. This locale boasts the Earth's oldest sediments, 3.5 or 3.8 billion years of age. So Carl Woese was the first to posit that the bacteria, archaea, and the eukaryotes represent separate evolutionary lines of descent that diverged at a very remote point in time on from an ancestral colony of organisms. So archaea and viruses likely had a relationship as early as 2 billion years before present day. Some researchers positing that the co-evolution may have been occurring between these groups at such an early time. It has been further suggested that the last common ancestor of the bacteria and archaea was a thermophile, raising the likelihood that low temperatures are really the extreme environments viewed from an ancestral archaea point of view, and organisms that can tolerate cold conditions appeared only later in evolutionary time. So next I am going to discuss about the taxonomy. It was not until 1977 that archaea were recognized as a separate domain of prokaryotes through the work of the Woese and Fox. Until 1965, the chief techniques of distinguishing microorganisms were use of the morphology and metabolic functions. So, Woese and Fox culminated a research direction begun by a number of researchers started in the early 1960s, in which gene coding of DNA material was viewed as a more fundamental technique for organisms' relatedness. By the close of the 20th century, an enhanced understanding of the significance and ubiquity of archaea arose by using the polymerase chain reaction to detect the prokaryotes in samples of the water or soil based solely upon their nucleic acid. The greatest remaining puzzle is whether to acknowledge the species within the domain of archaea, while the morphological and DNA findings support the recognition of species. It is not clear that significant gene transfer is prohibited, thereby annihilating the validity of the species. In any case, in the present treatment, we shall allow the attribution of species, if for no other reason than to follow the published research designations and for simplicity of naming. Next, I am going to discuss about the habitats of archaea. Archaea are microorganisms that define the limits of life on Earth. They were originally discovered and described in extreme environments such as the hydrothermal vents and terrestrial hot springs. They were also found in a diverse range of highly saline, acidic, and anaerobic environments. So this is archaea at the Midway Giza Basin, Yellowstone National Park, as shown into the figure. Next is further information about the habitats of archaea. So although many of the cultured archaea are extremophiles, these organisms in their respective extreme habitats represent only a minority of the total diversity of the archaea domain. The majority of archaea cannot be cultured within the laboratory setting, and their ubiquitous presence in the global habitats have been realized through the use of culture-independent techniques. One commonly used culture-independent technique is the isolation and analysis of nucleic acids, for example DNA and RNA, directly from the environment. So rather than the analysis of the cultured samples isolated from the same environment, culture-independent studies have shown that archaea are abundant and fulfill important ecological roles in cold and temperate ecosystems. Uncultivated organisms in the subdivision Crenarchaeota are postulated to be the most abundant ammonia-oxidizing organisms. In soils and to account for a large proportion, roughly 20% of the microorganisms present in the picoplankton in the world's oceans. In the subdivision of Eukaryote, uncultivated organisms in the deep sea marine sediments are responsible for the removal of methane, a potent greenhouse gas, via anaerobic oxidation of methane stored in these sediments. In contrast, uncultivated methanogenic methane producing Eukaryote from terrestrial anaerobic environments such as the rice fields are estimated to generate approximately 10 to 25% of global methane emissions. The cultured representatives of the Archaea are from high temperature environments such as the hot springs and submarine hydrothermal vents. Likewise, the cultured members of the Archaea include the organisms isolated from the hot environments. So organisms that are methanogenic and the organisms that grow vigorously in high salt environments, which are called as halophiles. So organisms in the Archaea lineage and the proposed Nanoarchaea lineage also inhabit the high temperature environments. However, the Nanoarchaea are highly unusual because they grow and divide on the surface of another archaea, which is called Ignicoccus. Nanoarchaea, which were discovered in 2002, contain the most smallest known living cell, 1/100th of the size of the Escherichia coli. The smallest known genome, 112 kilobases. So 1 kilobase is equal to 1000 base pairs of DNA. For comparison, the human genome contains 3.2 billion base pairs. So members of Archaea and Nanoarchaea have not been detected in pure culture. Rather, they have been detected only in mixed laboratory cultures. So archaea are also found living in association with eukaryotes. For example, methanogenic archaea are present in the digestive systems of some animals, including humans. So some archaea form symbiotic relationships with the sponges. In fact, Synarchaeum symbiosum was grown in a laboratory with its host sponge and was the first non-thermophilic Archaea to be cultured and described. It was the first organism which is considered for classification in the proposed Archaea lineage. So next I am going to discuss about the characteristics of the archaea. So although the domains bacteria, archaea and eukarya were founded on genetic criteria, biochemical properties also indicate that the archaea form an independent group within the prokaryotes and that they share the traits with the both bacteria and the eukaryotes. So major examples of the traits include: this is the cell wall of bacteria, which contains the peptidoglycan. So this is the cell membrane. This is peptidoglycan, this is outer membrane, and this is cytoplasm as shown into the figure. So first characteristic is the cell wall. Virtually all bacteria contain peptidoglycan in their cell walls. However, archaea and eukaryotes lack the peptidoglycan. Various types of cell walls exist in archaea. Therefore, the absence or presence of peptidoglycan is a distinguishing feature between archaea and bacteria. So this is the bacteria, and eukaryotes produce the membrane lipids consisting of fatty acids linked by ester bonds to a molecule of glycerol. So this is a glycerophospholipid, this is phosphate group, and this is glycerol, which is linked to a fatty acid chains as shown into the figure. So next characteristic is the fatty acids. Bacteria and eukaryotes produce membrane lipids consisting of fatty acids linked by ester bonds to a molecule of glycerol. In contrast, the archaea have the ether bonds connecting the fatty acids to molecules of glycerol, although a few bacteria also contain ether-linked lipids. No archaea have been discovered that contain ester-linked lipids. So the third factor is the complexity of RNA polymerase. Transcription within all types of the organisms is performed by an enzyme which is called as RNA polymerase, which copies a DNA template into an RNA product. Bacteria contain a simple RNA polymerase consisting of 4 polypeptides. However, both archaea and eukaryotes have multiple RNA polymerases that contain multiple polypeptides. For example, the RNA polymerases of the RNA contain more than 8 polypeptides. The RNA polymerases of eukaryotes also consist of a high number of polypeptides, which are 10 to 12, with the relative sizes of polypeptides being similar to that of hyperthermophilic archaeal RNA polymerase. Therefore, the archaeal RNA polymerases more closely resemble the RNA polymerases of eukaryotes rather than those of bacteria. So fourth characteristic is the protein synthesis. So various features of the protein synthesis in the archaea are similar to those of eukaryotes but not of bacteria. A prominent difference is that bacteria have an initiator, transfer RNA that has a modified methionine, whereas the eukaryotes and archaea have an initiator transfer RNA with an unmodified methionine. So next characteristic is the metabolism. So various types of metabolism exist in both bacteria and archaea that do not exist in eukaryotes, including nitrogen fixation, denitrification, chemolithotrophy, and hyperthermophilic growth. So methanogenesis, the production of methane as a metabolic byproduct, occurs only in the domain archaea, specifically in the subdivision of Urea archaeota. So classical photosynthesis using the chlorophyll has not been found in any archaea. The metabolic strategies utilized by the archaea are thought to be extraordinarily diverse in nature. So for example, the halophilic archaea appear to be able to thrive in high salt environments because they house a special set of genes encoding enzymes for a metabolic pathway that limits the osmosis. The metabolic pathway known as the methyl aspartate pathway represents a unique type of the anaplerosis, the process of replenishing supplies of metabolic intermediates. In this instance, the intermediate is methyl aspartate. Halophilic archaeans, which include the Haloarcula merismorti, a model organism which is used in the scientific research, are thought to have acquired the unique set of genes for the methyl aspartate pathway via a process which is known as horizontal gene transfer, in which the genes are passed from one species to another. So this is archaea as shown into the figure. While archaeans indeed look and appear like most bacteria, they are biochemically and genetically distinct from them. So scientific studies reveal that they are even closer to the eukaryotic counterparts rather than prokaryotic ones. One of the most distinguishing traits of these organisms is their ability to inhabit some of the most extreme environments. Hence, they are sometimes referred to as extremophiles. Some of them thrive in hydrothermal vents in the deep sea ocean at the temperature of over 100 degrees Celsius high. Some of them live in either extraordinarily acidic or basic waters, while the some are found to inhibit the digestive tracts of animals, while the some of them even live in petroleum deposits underground. In terms of reproduction, archaeans undergo asexual reproduction through binary fission, meaning the genetic material, which is DNA, duplicates as the cell wall pinches off the cell. So next I am going to discuss about the intermediate characteristics of archaea. The members of the domain Archaebacteria possesses a curious mix of the characteristics of prokaryotic and eukaryotic organisms. The table below showcases some of the intermediate traits. So first I will discuss about the prokaryotic traits and then I will discuss the eukaryotic traits. Prokaryotic traits: circular chromosome. Eukaryotic traits: DNA contains histones and other nucleosome-like structures. Prokaryotic traits: has oprons but lack introns. Eukaryotic traits: mechanism for DNA replication, transcription and translation. Prokaryotic traits: able to perform the metabolic functions like the energy generation, synthesis of polysaccharides and nitrogen fixation. Eukaryotic traits: has the RNA polymerase for transcription and initiation and elongation factors for translation. However, archaeans have some genes that are not like any found in anything else. For instance, the archaeans that thrive in extremely saline environments have unique defensive molecules and mechanisms for breaking the cells out. Another thing is that some archaeans have molecules that come in contact with the acids in order to help them preventing from the bursting. So next I am going to discuss about the structure of archaea. Archaea are prokaryotes, which means that the cells don't have a nucleus or any other membrane bound organelles in the cells. Like bacteria, the cells have a coiled ring of DNA and the cell cytoplasm contains the ribosomes for the production of cell proteins and the other substances the cell needs. Unlike bacteria, the cell wall and membrane can be stiff and give a cell a specific shape, such as the flat, rod-shaped or cubic. So archaea species share common characteristics such as the shape and the metabolism, and they can reproduce via binary fission just like the bacteria. So horizontal gene transfer is common. However, the archaea cells may take up plasmids containing DNA from their environment or exchange DNA with other cells. As a result, archaea species can evolve and change rapidly. So next is the cell wall. The basic structure of the archaea cell walls is similar to that of bacteria in that the structure is based on carbohydrate chains. Because archaea survive in more varied environments than other life forms, the cell wall and cell metabolism have to be equally varied and adapted to their surroundings. As a result, some archaea cell walls contain the carbohydrates that are different from those of bacterial cell walls, and some contain the proteins and lipids to give them strength and resistant to chemicals. So next character is the cell membrane. So some of the unique characteristics of archaea cells are due to the special features of their cell membrane. The cell membrane lies inside the cell wall and controls the exchange of substances between the cell and its environment. Like all other living cells, the archaea cell membrane is made up of phospholipids with fatty acids chains. But the bonds in the archaea phospholipids are unique. All cells have a phospholipid bilayer. But in archaea cells, the bilayer has ether bonds, while the cells of the bacteria and eukaryotes have ester bonds. Ether bonds are more resistant to chemical activity and allow the archaea cells to survive in extreme environments that would kill the other life forms. While the ether bond is a key differentiating characteristic of archaea cells, the cell membrane also differs from that of the other cells in the details of its structure and its use of long isoprenoid chains to make its unique phospholipids with fatty acids. The differences in the cell membranes indicate an evolutionary relationship in which the bacteria and eukaryotes developed subsequent or to us separately from archaea. Next is the genes and the genetic information. Like all the living cells, archaea rely on the replication of DNA to ensure the daughter cells are identical to the parent cell. The DNA structure of archaea is simpler than that of eukaryotes and similar to the bacterial gene structure. The DNA is found in the single circular plasmids that is initially coiled and that straighten out prior to the cell division. While this process and the subsequent binary fission of the cells is like that of bacteria, the replication and translation of DNA sequences takes place as it does in eukaryotes. Once the cell DNA is uncoiled, the RNA polymerase enzyme that is used to copy the genes is more similar to eukaryote RNA polymerase than it is to the corresponding bacterial enzyme. So creation of the DNA copy also differs from that of bacterial process. DNA replication and translation is one of the ways in which the archaea are more like the cells of animals than those of bacteria. So next character is the flagella. As with bacteria, flagella allows the archaea to move. Their structure and operating mechanisms are similar in archaea and bacteria, but how they evolved and how they are built differ. These differences again suggest that archaea and bacteria evolved separately with the point of differentiation early on in evolutionary terms. Similarities among the members of the two domains can be traced to later horizontal gene or DNA exchange between the cells. The flagellum in archaea is a long stalk with a base that can develop a rotary action in conjunction with the cell membrane. The rotary action results in the whip-like motion that can propel the cell forward. In archaea, the stalk is constructed by adding the material at the base, while in bacteria, the hollow stalk is built up by moving the material up the hollow center and depositing it at the top. So flagella are useful in moving the cells toward food and in spreading out after the cell division. So these are the three domains of life. So first is the bacteria, which includes the E. coli. Next is archaea, which includes thermophiles. And the third one is eukaryota, which includes animals, fungi and plants as shown into the figure. So next I am going to discuss about the general morphology. Archaea and bacteria are superficially similar in size and shape. Although some archaea species have remarkable geometric shapes, such as the flat and square-shaped cells of some genus like the halo quadra members. Despite this visual similarity to bacteria, archaea possesses the genes and several metabolic pathways that are more closely related to those of eukaryotes, notably the enzymes involved in gene transcription and translation. So other aspects of the archaean biochemistry are unique, such as the occurrence on the ether lipids within their cell membranes. As with bacteria, archaea have no interior membranes or organelles. Cell membranes are typically bounded by a cell wall. Motility is achieved using one or more flagellar tail structures. So archaea most resemble gram-positive bacteria. So most archaea exhibit a single plasma membrane in cell wall, lacking a periplasmic space. However, Ichthyococcus manifests a notably large periplasm with membrane-bound vesicles enclosed by an outer membrane. Certain archaea aggregate to yield the filaments of the cells as long as 200 nanometers. Such forms are prominent in biofilms. Thermococcus coalescens. On the other hand, have the cells that can fuse in culturing to produce the monster single cells. Genus Pyrodicticum or Pyrodictium. Archaea form an elaborate multi-cell colony manifesting arrays of slender, elongated hollow tubes termed cannulae that protrude from the cellular surface and connect into a dense agglomeration. This protruding form appears to encourage the connection or nutrient exchange with the neighboring cells of the same genus. So Crenarchaeota exhibit a diverse set of geometries, irregularly shaped lobed cells, needle-like filaments that are less than 500 nanometers in cross-section, and amazing rectangular rods. So these odd morphologies are likely produced both by their cell walls as well as a prokaryotic cytoskeleton. So proteins associated with the cytoskeleton elements of the other organisms exist in archaea. So next is flagella. So archaea flagella function like their bacterial counterparts with elongated stalks driven by the rotatory motors at the base. The motors themselves are powered by the electrochemical gradient across the membrane. So however, archaeal and bacterial flagella came from different ancestors. The bacterial flagellum is hollow and is assembled by subunits moving up the central pore to the tip of the flagella, while archaeal flagella are constructed from the addition of subunits as the base. So next is the membranes. The membranes of archaea are constructed from molecules unlike those in other life forms. This morphology demonstrates the ancestral distance from bacteria and eukaryotes. For every organism, the cell membranes are made up of phospholipid molecules. These phospholipids exhibit a polar part that dissolves in water, a phosphate head, and a hydrophobic nonpolar part, a lipid tail, that is water insoluble. These dissimilar ends are connected by the glycerol group. In water, the phospholipids aggregate with heads facing the water and tails facing the opposite direction. The principal structure in cell membranes is the dual layer of phospholipids, often termed as the lipid bilayer. These phospholipids are distinct for a number of reasons. In the case of bacteria and eukaryotes, the membranes consist chiefly of a glycol ester lipids, but archaea have membranes made of glycerol ether lipids. So ether bonds are chemically more stable than ester bonds, assisting the archaea in survival at extreme temperatures and extreme pH environments. So stereochemistry of the glycerol group is the reverse of that found in other organisms. This implies that the archaea use a wholly different enzymes for synthesizing the phospholipids than bacteria and eukaryotes. So such enzymes developed in a very ancient geological times, suggesting an early split from the other two domains. In the some archaea, the lipid bilayer is replaced by the monolayer, in which the tails of the two independent phospholipid molecules are fused into a single molecule with the two polar heads. This fusion may make their membranes more rigid and better able to resist the harsh environments. Phaeroplasma is an example. This organism's survival in its highly acidic habitat is abated by the such tail fusion. So archaea lipid tails are chemically distinct from the other organisms. Archaea lipids are based upon the isoprene side chain. They are long chains with the complex side chains and often cyclopropane and cyclohexane rings. Although isoprenoids play an important role in the biochemistry of many organisms, only the archaea use them to make the phospholipids. These branch chains may aid the archaean members from leaking at high temperatures. So next is the metabolism. Many fundamental metabolic pathways are shared between all forms of life. So as a case in point, archaea invoke a modified form of glycolysis and either a complete or partial citric acid cycle. These likenesses to the other organisms reflect archaean early origins as well as the archaean high metabolic efficiency. So a vast variety of chemical reactions are applied by the archaea in supporting their metabolic behavior, so employing a host of energy resources or energy sources. These reactions can be grouped into nutritional groups. In some cases, the inorganic compounds, for example ammonia and sulfur, supply the energy to the archaeans, which is known as the lithotrophs. The other set of archaea employ the sun's energy while not engaging in actual oxygen producing photosynthesis. This archaean group is known as the phototrophs. No archaea are known to use the photosynthesis. Phototrophic archaea use the sun's energy to produce the chemical energy in the form of ATP. In the halobacteria, the light activated ion pumps like the bacteriorhodopsin produce the ion gradients by the pumping ions out of the cell through the plasma membrane. The energy stored in such electrochemical gradients is subsequently converted into ATP by the ATP synthase in a process that is a form of photophosphorylation. The ability of these light-driven pumps to transport the ions across the membranes depend on the sunlight-driven alterations in the structure of retinol cofactor embedded in the protein center. So some swamp dabbling archaea thrive in anaerobic settings. In fact, this primitive form of metabolism may have powered the first free living organism. Such methanogenic metabolism relies upon the carbon dioxide as an electron acceptor to oxidize the hydrogen. So methanogenesis invokes a gamut of the coenzymes unique to these archaea, including the coenzyme M and methanofuran. So sometimes the various alcohols and acetic or formic acid are employed as in methanogenic electron acceptors. So these reactions are commonly used in the intestine dabbling archaea. So acetic acid is also decomposed into methane and carbon dioxide by the acetotrophic archaea. So these acetotrophs are archaea in the order of the methanosarcinales and are a major part of the ecological microorganism communities that produce the biogas. Atmospheric carbon is another source of energy input for autotrophic archaea who employ the carbon fixation. This process involves either first is a highly modified form of Calvin cycle or second is a metabolic pathway which is termed as the 3-hydroxypropionate. By 4-hydroxybutyrate cycle, 3-hydroxypropionate divided by the 4-hydroxybutyrate cycle. So next is the reproduction. Having no cell nucleus, so archaea do not produce via mitosis. Rather, they procreate using a process which is also known as binary fission. In this binary fission process, the archaeal DNA replicates and the two strands are pulled apart as the cell grows. In some cases, the more than two daughter chromosomes can be created and subsequently pulled apart in a process which is called as multiple fission. So archaeal chromosome replicate from multiple origins of replication using DNA polymerases that resemble the counterpart eukaryotic enzymes. However, the proteins that direct the cell division, such as the protein which is called as the FTSZ that forms a contracting ring around the cell, and the components of the septum constructed across the center of the cell resemble the bacterial likenesses. So Anion Walk et al. Demonstrated that the archaea, unlike many bacteria and the eukaryotes, produce no spores. Next I am going to discuss about the habitat. So it was the first thought that the most archaea were extremophiles, existing at the environmental limits of abiotic factor ranges. Recently it has been found that there are numerous archaea living in a broad range of habitats and environmental conditions. Some haloarchaea undergo the phenotypic switching and grow several different cell types, including the thick walled structures that are highly resistant to osmotic shock. These thickened walls permit the survival under the hyposaline, low salt circumstances. But these alternative phenotypes are not actual reproductive structures. Rather, they may assist the archaea in reaching the new habitats. Indeed, some archaea thrive in extreme temperatures, often above 100 degrees Celsius. For example, they occur in hot springs and geysers, black smokers, and oil wells. Other viable environments include very cold environments and highly saline, acidic, or alkaline media. For example, the Picrophilus torridus, an extreme Archean acidophile, thrives at pH of essentially 0, equivalent to a 1.2 molar concentration of sulfuric acid. So archaea are also found in the very cold ocean environments, including the polar seas. Many archaea also occur throughout the world's oceans among the plankton communities as a part of the picoplankton. Moreover, archaea include the mesophiles that grow in mild conditions, in marshes, sewage, the oceans, and soils. Halophiles, including the genus Halobacterium, survive in the hypersaline environments such as the salt lakes and can outcompete bacterial counterparts at the salinities greater than 20%. The thermophiles grow best at the temperatures above 45 degrees centigrade. In locales such as the hot springs, hyperthermophilic archaea grow optimally at the temperatures greater than 80 degrees Celsius. Strain 166 of the Archean Methinopyrus candleri survives at the 122 degrees centigrade Celsius. 122 degrees Celsius, the highest recorded temperature for any organism. So this is a color transmission electron micrograph, which is also known as TEM, of a section through the Archaebacterium, which is known as the Methinococcoides burtoni or burtoni, as shown into the figure. So next I am going to discuss the three types of Archaebacteria. The Domain Archaea is a group of unusual organisms and is known to include the three groups, which are methanogens, extreme halophiles, and thermoacidophiles. So first I am going to discuss about the methanogens. As their name suggests, methanogens are archaeans that are capable of producing methane gas. And this amazing ability to produce the methane makes them easily distinguishable in the digestive tract intestines. These organisms are found in environments that have no oxygen, like the anoxic, in muddy marshes and the swamps, and the digestive tracts of the cows and termites and marine lives. The methanogens get their energy from the utilization of hydrogen and carbon dioxide (CO2). At present, the two methanogens have already had their complete sequenced genome, which includes the Methinocladococcus genetiae and the Methinothermobacter thermoautotrophicus. The chemical equation for methane gas is 4H2 plus CO2 is equal to CH4 or marsh gas plus 2H2O. So this is the methane which is producing from the cow's bobs as shown into the figure. Next is the extreme halophiles. So halophiles, coming from the Greek word halos, which means the sea or salt, are a type of the archaea that inhabit the extremely salty environments like the Dead Sea or the Great Salt Lake in Utah. Interestingly, these extreme halophiles produce a pigment which is known as the bacteriorhodopsin that gives them the ability to utilize the sunlight as a source of energy. In the same manner, plants use it for the photosynthesis. Aside from the sea, the halophiles can also live in the soil as long as the salt level is very high. So this is the Great Salt Lake in Utah, as shown into the figure. So third type is the thermoacidophiles. Thermoacidophiles, as their name suggests, are archaea that thrive in both hot and acidic environments, especially in those near the sulfur hot springs. The ideal temperature and pH levels for the thermoacidophiles are at 70 degrees Celsius and higher, and pH levels of 1 or 2 similar pH as concentrated sulfuric acid. However, when subjected to lower temperatures below 55 degrees Celsius, 131 degrees Fahrenheit, they die. Despite this, it is essential to note that not all archaea are extremophiles. Many of them thrive in normal environmental conditions. In particular, some of them can be found alongside with the bacteria and some marine plants in the ocean. The main differentiating characteristic of archaea is their ability to survive in toxic environments and extreme habitats. Depending on their surroundings, the archaea are adapted with regard to their cell wall, cell membrane, and metabolism. So archaea can use a variety of energy resources, including the sunlight, alcohol, and acetic acid, ammonia, sulfur, and carbon fixation from the carbon dioxide in the atmosphere. The waste products include the methane, and methanogenic archaea are the only cells able to produce this chemical. The archaea cells able to live in extreme environments can be classified depending on their ability to live in specific conditions, such as four such classifications are: tolerance for high temperatures are called as hyperthermophilic, able to survive acidic environments are called acidophilic, can survive in highly alkaline liquids which are called as alkaliphilic, tolerance for high salt content are known as halophilic. So some of the most hostile environments on the earth are the deep sea hydrothermal vents at the bottom of the Pacific Ocean and hot springs such as those found in Yellowstone National Park. High temperatures in combination with the corrosive chemicals are usually hostile to life, but archaea such as the Ignicoccus have no trouble with those locations. The resistance of the archaea to such conditions has led the scientists to investigate whether the archaea or similar organisms could survive in space or no otherwise hostile planets such as the Mars. With their unique characteristics and comparatively recent emergence to prominence, the archaea domain promises to reveal more interesting characteristics and capabilities of these cells, and it may offer the surprising revelations in the future. So next I'm going to discuss the importance of archaea or commercial uses. Because of their tolerance to high temperatures and relatively extreme temperatures, so some members of the domain have already been exploited for a wide variety of commercial uses. These archaea become the source of enzymes that are usually added to the detergents in order to help it maintain its activity even at higher temperature and pH. Some enzymes from archaea are also used to convert the cornstarch into the fiber dextrin. So some archaea also bear the potential for the bioremediation or help in cleaning the contaminated sites. The thermophilic archaea, which is known as Thermus aquaticus, is an essential part of the development of molecular biology as a science. As a result, archaean has become the source of the enzyme which are harnessed as the basis for the amplification of the DNA in a technique called the polymerase chain reaction or PCR. At present, members of the domain archaea may be the only organisms that can survive in extreme habitats. So much is still to be discovered about these microorganisms, and as the advancement of the technology becomes more extensive, the understanding of the microscopic world becomes the more comprehensive as well. So these are the archaeans in detergents as shown into the figure. Next I am going to discuss about the classification of archaea. So this is the classification of archaea. Based on various molecular and genetic compositions, the domain archaea is subdivided into 5 phyla that includes the Eukaryota, Crenarchaeota, Prokaryotic, Archaea, and Nanoarchaea. So first I will going to discuss about the Eukaryotic. The phylum Eukaryotic is one of the best studied phyla of the domain Archaea, consisting of more than 70 genera. Members of the phylum are extremely physiologically diverse with the ability to survive some of the most extreme environments around the world. The following are the characteristics of the different groups of the phylum Eukaryotic. So Eukaryotic is composed of mesophilic, thermophilic and cyclotolerant species spread across the 8 classes. So first class is the Archaeoglobi. The class Archaeoglobi is made up of a single order which is called Archaeoglobales, and the family which is known as Archaeoglobaceae or Archaeoglobaceae. Archaeoglobaceae is further divided into the 3 genera that includes the Phaeroglobis, Geoglobis and Archaeoglobis. Like class Thermocoxy, the members of Archaeoglobi are spherical in appearance and thus may be described as having a coccoid morphology. So some of the main characteristics of this group include found in hydrothermal vents in deep sea, anaerobic, grow well in marine salinity. And the 3 genera in the phylum contain species that are mixotrophic, use the different sources of energy, and lithoautotrophic, that obtain the energy from reduced compounds in nature. So optimum temperature range for the members of this phylum is between 83 and 88 degrees Celsius. Maximum temperature is about 95 degrees Celsius. Next is the class Protoarchaea, also known as the class Thermocoxy. Protoarchaea is also made up of a single order which is called Thermococcales, and the family which is called the Thermococcaceae or Thermococcaceae. The family Thermococcaceae is further divided into the genera Pyrococcus, Thermococcus and Paleococcus. With the exception of a few species, a majority of species in these genera have the following characteristics: obligate anaerobes, hyperthermophilic that thrive in extremely hot environments, organotrophic, grow well in neutral pH and marine salinity, and coccoid in morphology, capable of movement, so found in hydrothermal vents. So some species are capable of fermentation. All are obligate organoheterotrophs, so they can obtain the energy and nutrition from organic compounds or from the other organisms. Next is the class Thermoplasmata. So class Thermoplasmata is made up of one order which is called Thermoplasmatales, and the 3 families. Members of this class have the following characteristics, which are extreme acidophiles that thrive under acidic conditions, aerobic in nature, they need air for growth, spherical or coccoid morphology. Majority of species are moderate thermophiles, they grow under moderate temperatures below the 40 degrees centigrade. So some species, for example the members of the family which is called Phaeroplasmaceae or Phaeroplasmaceae, can oxidize the iron compounds to obtain the energy, so they are incapable of fermentation. So next class is the Halobacteria, consisting of about 30 genera. The class Halobacteria is made up of organisms that are highly halophilic in nature. As such, they are able to thrive well in the environments with a high salt concentrations, 30 to 36% sodium chloride. For this reason, they can be found in such extreme environments such as Dead Sea. So some of the other important characteristics of these members of this class include aerobic environments, aerobic organisms that need the air for growth. Many of the species also need the magnesium salts for their development. So most of the species are paleomorphic in morphology, which is caused by the high minerals in their environments. A majority of the species are gram-negative, with only a few being gram-variable. Mesophilic and chemoorganotrophs. They oxidize the chemical bonds of complex organic compounds like the peptone, etc. to obtain the energy. A majority of the species are found in such salty environments as soda lakes and marine springs containing the sulfur and sulfide among others. Next is the methanogens. So methanogens encompass four classes of the phylum Eukaryotic that are characterized by their ability to produce the methane. So these include the methanotherma, methanobacteria, methanopyri, and methanomicrobia. So apart from the ability to produce the methane, all the members of this group are obligate anaerobes that use the carbon dioxide to accept the electrons. As such, they cannot tolerate the presence of oxygen. Some of the other characteristics of methanogens can be found in the digestive system of the ruminant animals, for example, the goats, domestic yak. In the rumen of these animals, the methanogens are involved in the fermentation process, are found in the deep sea and saline environments, for example, the members of class methanopyri and methanothermia. Vary from the coxy and curved rods to long and short in morphology. Grow well in neutral environments or slightly alkaline environments. So this is the archaea by the Demasteridae as shown into the figure. Next is the phylum Crenarchaeota. Apart from the phylum Eukaryotica, phylum Crenarchaeota is the other group of organisms that has received a lot of attention over the years. Although this phylum contains fewer genera compared to the former, it consists of a great diversity of organisms that can be found in various types of environments. For instance, whereas some of the species can be found in the soil, others can be found in high temperature environments like the thermophiles. And compared to the phylum Eukaryotica, a single class Crenarchaeota of the phylum Crenarchaeota has been identified so far. So Crenarchaeota is further divided into 5 orders that include the order Acidobales. So the Acidolobales. So members of this order are acidophiles that use the sulfur during respiration as the electron acceptor. They are spherical in the shape, coxy, and include the members of two major families, namely Acidilobaceae or Acidilobaceae, and next is the Cladiscipheraceae or Cladiscipheraceae. Next order is the Desulforococales. This order is divided into the family Pyrodictaceae or Pyrodictaceae and the family Desulforococcaceae or Desulforococcaceae. Whereas some are coxy in their morphology, the majority of the Desulforococcaceae, others are rod-shaped. So some of the other characteristics of the order Desulforococcales include: some are obligate anaerobes in nature, Desulforococcaceae. Slightly acidophilic or neutrophilic organisms that can be found in some marine environments, Desulforococcaceae. So some members are fermenters, and some members of this family, Pyrodictaceae, are thermophiles and that can survive the extremely high temperatures. G plus C content varies from one group to another. Next order is Sulfolobales. So the order Sulfolobales consists of a single family which is known as the Sulfolobaceae or Sulfolobaceae, which is in turn made up of hyperthermophilic and acidophilic organisms spread across the 6 genera. So characteristics of this order Sulfolobaceae vary from one group of organisms to another. For instance, whereas the genus Sulfurisphera is made up of an organism that is strictly organotrophic and facultative anaerobe, members of the Acidianus and Sulfolobus among the others have been shown to use the lithoautotrophic and organotrophic metabolism. Next order is the Thermoproteales. This order is composed of 2 families, which is Thermophyllaceae or Thermophyllaceae and the Thermoproteaceae or Thermoproteaceae that have the following characteristics. which are neutrophiles or slightly acidophilic organisms that can be found in the high temperature environments, and hyperthermophiles. Rod-shaped, some are fermenters or thermoproteus. Some can grow on compounds containing hydrogen and thiosulfates, etc. Organoheterotrophs. Next order is the Furvidicoccales. This order consists of a single family in species that can be found in hot springs. The other three phyla of the Archaea are not fully understood and no valid representatives have been agreed on. The following are some of the characteristics of the three groups. The phylum Prokaryotic. This phylum was discovered in both marine and terrestrial hot environments, and thus suggesting that members of the phylum are hyperthermophilic. In order to determine the diversity and abundance of the groups in nature, studies have been conducted in various environments and in a number of the countries. Through these studies, it became evident that the members of phylum grow in environments that range between 70 to 97 degrees Celsius in temperature and 2.5 to 6.5 in pH. Currently, very few organisms have been identified as belonging to the phylum. So, one example of this is the Candidatus Coraceum cryptophyllum that was isolated from a culture containing the sediments from the obsidian pool. So, based on these studies, the organisms, the following characters were identified: capable of degrading the peptides through fermentation, cannot synthesize such cofactors as vitamins. Next is the Nanoarchaea. Like the phylum Prokaryotic, only one member, Nanoarchaeum equitans of the phylum Nanoarchaea, has currently been identified. So N equitans have been shown to grow attached in a symbiotic relationship to the surface of various Ignicoccus species that have the following characteristics. Hyperthermophilic organisms can be found on the hot rocks, marine thermal vent environments and springs. Are strict anaerobes, contain divergent 16 subunit ribosomal DNA sequence, lack genes responsible for the synthesis of lipid or amino acids and nucleotide, grow well in high temperatures about 90 degrees Celsius and pH range of between 2.5 and 3.0. Next is the Thymarchaea. As compared to Prokaryotic and Nanoarchaea, Thymarchaea is better understood, with the group making about 5% of all the prokaryotes in the soil system. They can also be found in hot springs and marine waters and consist of ammonia oxidizing organisms. So some of the main characteristics of Thymarchaea include a majority of the organisms that can fix the carbon dioxide under the autotrophic conditions. They oxidize ammonia aerobically. So next I am going to discuss about the Archaea groups. Apart from the phylum divisions of Archaea, the domain is also divided into the following three groups. First is extreme halophiles. Extreme halophiles include a variety of organisms that thrive in an environment that contain the high salt concentrations. For optimal growth, extreme halophiles have been shown to require at least 1.5 mol per liter of sodium chloride. Therefore, while many may tolerate the high salt conditions, a good number have been shown to actually depend on such conditions for growth. Examples of extreme halophiles include Haloadaptus, Halobaculum, Methanohalobium. So, next is the methanogens. Methanogens are characterized by the inability to tolerate the oxygen as well as the ability to produce methane. So methane gas is therefore produced under anaerobic conditions and in the absence of such ions as ferric ions and nitrates. In cases where the methanogens live in anoxic soils or in environments where the other organisms produce oxygen, they produce methane at a high rate in order to bring about anoxic conditions. Because of their ability to produce the methane, they have been used in some industries to produce the gas. Examples of methanogens include the Methanopyrus candleri, Methanocladococcus, and next is the Methanobrevibacter. Next is Methanosphera, Next, I am going to discuss about the hyperthermophiles. So hyperthermophiles are also commonly referred to as the heat-loving prokaryotes. This is because they are a group of archaea that are capable of growing in temperatures of above 80 degrees Celsius. They are commonly found in environments with a very high temperature, such as the hot acid springs in geothermal power plants, as well as submarine volcanic habitats and areas with heated soil. So apart from the high temperatures, some hyperthermophiles have also been shown to tolerate extreme acidity in their environments. So however, the majority, which is obligate anaerobes, grow well in the environments that are either neutral or mildly acidic. So examples of the hyperthermophiles include Geoglobus, Archaeoglobus and Ferroglobus. Next, I am going to discuss about the main characteristics of archaea versus bacteria. Main characteristics of archaea versus bacteria. Because of their diversity, the archaea cells display the significant variance in morphology. Whereas the some are rod-shaped like many bacteria, others are spiral, disc-shaped or spherical in shape. On the other hand, some have been shown to portray various irregular shapes. Although archaea is a distinct domain, it shares a number of the characteristics with both bacteria and eukaryota. For instance, like bacteria, a majority of archaea have a cell wall that regulates the osmosis and maintains the shape of the cell. However, unlike bacteria, the archaea do not have the peptidoglycan. Rather than they contain the pseudopeptidoglycan consisting of N-acetyl talosamine uronic acid or NAT, while the others have the cell wall which is made up of proteins or polysaccharides. Both bacteria and archaea are also capable of locomotion in moist or liquid environments. This is made possible by the presence of flagella. Depending on the species, archaea may possess one or several flagella, allowing it to move from one point to another. Although they possess the flagella that allow for locomotion, the flagella protein and the structure of flagella is different between the two. The other difference between the archaea and bacteria is with regards to their cell membrane. While both have a cell membrane, there is a difference in how various the components of the cell membrane are arranged in archaea. For instance, in archaea, the tails of the hydrophobic lipid are attached to the glycerol by the ethyl linkages. This is the different from the ester linkage which is present in bacteria and eukaryota. With both the bacteria and archaea being prokaryotes, the chromosomes region is composed of bodies which is known as nucleoids. Compared to the eukaryotes, these diffuse mass lack a membrane envelope and thus reside in the cytoplasm as DNA aggregates. Both bacteria and archaea contain the plasmids, small DNA molecules. These extra chromosomal molecules of DNA are typically circular in shape with the genes ranging from about 5 to 100. Like eukaryotes, bacteria and archaea also have been shown to have a cytoskeleton that regulates the cell division. Next, I am going to discuss about the interesting archaean facts. While archaeans are very similar to bacteria, they are also much different. Unlike some types of bacteria, the archaeans cannot perform photosynthesis. Similarly, they can produce spores. Archaeans are extremophiles and they can live places where the most other life forms cannot. They can be found in extremely high temperature environments as well as the extremely low temperature environments. So archaeans are a natural part of the human microbiota. So at present, the pathogenic archaeans have not been identified. Scientists assume that they do not exist. If you like this video, please subscribe to my YouTube channel and press the bell icon as well for further updates and notifications and information. Thanks for watching. Thank you so much for your time and appreciation.